let's look at the arrangement of photoreceptors within the retina and explore the significance of that arrangement for the functioning of the system. Here's an en face view of a primate retina. We see each of these white spots is a single rod photoreceptor cell. These are the closely packed ones. And the clear spaces with a single white spot within it are the cone photoreceptor cells. You can see that it's essentially a sea of rods with cones interspersed like little pebbles. Now this is what the peripheral retina looks like, the region that's off to the side. But the retina overall is not a homogeneous structure with respect to photoreceptor topography. In the primate retina, including the human retina, if we were to map across the width of the retina from one side to the other, the density of the different photoreceptor cell types, we would see a very distinctive distribution. So for example, the rod photoreceptors are very abundant, that's these dots here, are very abundant in the peripheral retina. The cone photoreceptors down at the bottom are relatively rare in the peripheral retina, as we saw on the previous slide. But towards the central retina, and especially at the very center of the retina, called the fovea, the cone density rises to a very high peak, and the rod density correspondingly goes down to essentially zero. That's a very striking arrangement. Why is that? It turns out that the retina of primates like ourselves is really a two-stage structure in the sense that we have a high acuity zone of the retina, this very central zone, which encompasses just a tiny fraction of the retinal surface area, less than 1%. And the rest of the retina really exists simply to alert the central retina that something interesting has happened. Whenever we look at something that we're interested in, what we do is we turn our eyes towards that object. And we do that because we want the object to be imaged at on the central region of the retina. Let's look at one simple consequence of this specialization within the retina, and that is the eye movements that allow different regions of the world in which we're interested in to be sequentially imaged on the central retina. And there's no better example than reading text. So if you're reading the page of, for example, a novel, and you look at the eye movements that correspond to that process, you'll see that as we look here on the horizontal axis at eye position and on the vertical axis at time, if we look at the way the eye moves, we'll see that it's a series of stops and starts where the eye is initially focused on one location, and then there's a rapid movement to a new location, a brief pause at that place, and then a rapid movement to yet another location, and so on, as we read from word to word across the page. And of course, when we get to the end of the line, then we reset all the way back to the beginning of the next line. Why are we doing that? Why can't we just stare straight ahead and read? And the answer is that when you're reading, you need to see the words, the letters of interest at high acuity. And the only way to do that is to have them fall on the very central retina. So your eye must continually be moving to allow the object of interest to fall in that most central region, the fovea. Really, the rest of the retina exists simply to get our attention and tell us about an object that we might want to image on the fovea. For example, if you see a fly in your peripheral vision buzzing around the room, that will grab your attention and you'll perhaps turn your eye towards the fly, but you won't really get a good look at it until you turn your eye towards it. Now let's look at eye movements in a somewhat more natural context. And this tells us also something not only about the eye, but about the brain. That is, about which regions of a given scene, perhaps a more natural scene than reading of text, grabs our attention. And so here, for example, we're looking at eye movements that correspond to looking at faces. So the top two photographs are the images that were observed, and the bottom data points indicate where the eyes were pointed during viewing of that image. So when looking at a face, for example, this young lady's face, you can see that the eyes of the observer tend to look at her eyes, And they also look at her mouth, they look at her nose a little bit, maybe around the face. If we look at a face in profile, again, you can see from where the eyes were pointing that they tend to look at the edges of the image, the nose, the eyes, the chin, the mouth, and so on, and largely neglect the central regions, the cheeks, the neck, and so on, that are perhaps of lower interest and where less is going on. This is a highly selective viewing strategy. And so it really means that we're sampling the world in a way that is far from random and far from complete. We're sampling just subsets of any given scene. Now, the fact that the central retina is the only place where the image is seen with high acuity means that if you're going to examine, say, a non-moving object, and you're doing it in the context of a head and a body that are moving, perhaps because you're walking or running or just trying to stay still, but you're not entirely still, there must be a mechanism that allows the eye to stabilize itself in the context of this moving holder, that is the rest of the body. And in fact, that mechanism exists, and it's a very powerful one. Here's an example just showing how that sort of movement compensates for head and body movements during walking. Again, on the horizontal axis, we have the angular degree, and we have plotted here in red the head movements associated with walking. Of course, there's some swaying back and forth with each step, and as time goes on, moving up this chart, we see that the head movements in red and the eye movements within the head in green are nearly mirror images of each other. That is, the eye is moving within the socket to almost perfectly compensate for the back and forth movement of the head. It's completely unconscious. We do it without thinking about it. But the result is that the eye is pointing nearly perfectly straight ahead during the entire walk by virtue of this moment-to-moment sense of where it is deflected. If it's deflected a little to the left, it'll move to the right. Deflected a little to the right, it'll move to the left. And this, of course, keeps the world looking relatively still, despite the fact that we are moving around. Now, let me say one final thing about the arrangement of photoreceptors within the retina, this non-random arrangement, and that now relates to the distribution of the different cone photoreceptors. We haven't talked about the different kinds of cones. We're going to talk in detail about that in the second and third lectures. But in the human retina, let's just, for the moment, note that there are three different cone classes, one most sensitive to longer wave light, one most sensitive to medium wavelengths of light, and one most sensitive to shorter wavelengths of light. The medium and longer wave receptors, and I'll call these M and L, are in fact rather similar to one another with largely overlapping absorbances, whereas the shorter wave, which I'll call S, is really quite different. It absorbs at wavelengths quite down towards the short end of the visible spectrum. And as a consequence, any given image which contains wavelengths throughout the visible spectrum and which therefore would be exciting all the different photoreceptor cells will, when it arrives at the retina, not be perfectly focused. Let's think about why that is. It's because any lens, including our own lens, has some degree of what's called chromatic aberration. That is, the image being focused for a given wavelength is not going to be focused perfectly for some other wavelength. And so, for example, as shown here in this upper image, if this word red in red letters is focused perfectly on the retina so that it's sharply focused and a point in the outer world comes to a point on the retina, then blue light coming from the same region of space would be focused actually ahead of the retina, so that the word blue here, as shown on the right, would be a bit blurry, a bit out of focus. How does the retina deal with this problem? It turns out that in the very central fovea, the S cones have been excluded. Only M and L cones populate that region. That's the region with the very highest acuity. And what the retina has done, what the eye has done, is simply eliminate the short wave end of the spectrum for that most high acuity zone of the retina. So by taking a narrow cut of wavelengths, we're effectively getting a sharper image, of course at the price of having a less colorful image. Now let's talk about how the image is processed in the retina. We mentioned at the very beginning of the lecture that the retina is really different from a film and a camera in the sense that it is not just detecting the image and sending that detected image to the brain, but it's processing it and extracting from it those aspects that are of greatest interest to the organism. Let's look at one of those feature extractors here, and this can be illustrated with this classic illusion, the so-called herring illusion. I hope you can see that at the intersections where these white streets come together, there are illusory black dots. Actually, not on the one that you might focus on, but on the ones that strike your peripheral retina. I'll leave it as a homework exercise to figure out why that should be so. But if you can't see these as perhaps clearly as I can, I invite you to produce an illusion of just this sort on a home computer. You can do it either with black squares and white streets in between, or the reverse. If you do it with white squares and black streets, you'll see little white dots at the intersections of the streets. And this illusion has been known for well over a century, and it turns out to be fully explained by a peculiar type of organization, spatial organization of the receptive fields of ganglion cells. Now let me define receptive field. That refers to that zone of primary photoreceptor input which influences the output of the retinal ganglion cell. The classic retinal ganglion cell receptive field is as shown here. There's a little zone of retina, so we're looking now en face at the retina. There's a little zone of retina where illumination excites the ganglion cell, would increase, for instance, the firing of action potentials. And then there's an annulus around it, like a donut, where light activation of photoreceptors actually inhibits the ganglion cell. This is essentially a contrast detector. The ganglion cell is looking for spatial differences in illumination in the retina. And let's see how this explains that illusion. Here I've shown superimposed on a little region of that so-called herring illusion, one of the receptive fields of a ganglion cell, where in the center, the ganglion cell is looking at the light that falls at one of these intersections between white streets. And I think we can see that for this particular cell, not only is the center being illuminated, which would tend to excite the cell, but a rather substantial chunk of the surround on the right and left sides, above and below, is also being illuminated. So the result would be that this cell would be substantially suppressed by that illumination of the inhibitory surround. What happens if we look at a ganglion cell that's a little off to the right? Its center is also fully illuminated, but now I think you can appreciate a bit less of the surround is illuminated than was the case for the first cell. But now if we look at a cell that is even further off to the right, where we see the center is illuminated, but now the surround is only minimally illuminated, just this horizontal region here is receiving light, I think we can appreciate that this cell would be substantially more active because the central region being fully illuminated is countered by only a modest amount of inhibitory illumination just from the left and right sides. So the brain would perceive, or the retina would perceive, this zone of white street as brighter relative to the zone over here, because the ganglion cells seeing the street that's off to the right or off to the left side would have greater overall activity than would the ganglion cell that was centered over this intersection here. And that is the source of that illusion of blackness, that sort of fuzzy blackness at the intersections between those streets. I should just say that this idea of center surround ganglion cells was really predicted by this illusion many years before those cells were actually identified electrophysiologically. Now this leads us to a larger question related to image processing in the visual system. And here we're going to move a little bit beyond the retina into the brain. But if what the retina and also the brain are looking for, among other things, are differences between one region of the image and another region, as illustrated by those center surround ganglion cells, then it stands to reason that schematics of the sort that bring out those differences might resonate in some way, might have a special meaning in terms of their information content for our visual system. And that is exactly the case. So for example, we see here on the left a photograph of a hand with fingers about to snap. And on the right, we see a schematic here, just a line drawing of the same kind of hand. The fingers are about to do like this. And we immediately recognize in this simple line drawing what's going on. But that's actually kind of surprising that we should recognize this image of a hand so readily. After all, on a pixel by pixel basis, this little schematic bears very little resemblance to the photograph. The schematic shows black lines that outline each object, each finger and the palm of the hand, but the real world isn't like that at all. In fact, the real world is made of shapes with shadows and complex changes in illumination as shown by that photograph, very different on a pixel by pixel basis from this schematic. Why is it the schematic is so immediately recognizable? I think almost certainly it's because that the schematic is essentially the processed version that our retina and our brain has created from the real image. We've fed the visual system by giving it this schematic, a version which is already partway up the visual chain of command. Let's see another example of this. Here in this self-portrait by Pablo Picasso, we see the remarkable effectiveness of simple portrait sketches. Why is this so immediately recognizable as a face? And in fact, it's recognizable as Picasso's face. Again, even though on a pixel by pixel basis it's very different from a real human face, it has abstracted the information in just the way that our brain abstracts it as well. Now, there are other things that the visual system is interested in besides changes over space, changes in intensity over space. It's also interested in changes over time and the combination of time and space. Let's look at an example of time changes. Here are the responses of a retinal ganglion cell in the rabbit retina to an object moving in various different directions. The directions of motion across the receptive field are indicated by the arrows in the center here. And the responses that go with each of those directions of motion are indicated by the cluster of action potentials recorded from that cell that are indicated adjacent to the corresponding arrow. Now, I think you can appreciate that motion in the upward direction and directions that are close to it are especially effective, but motion in the downward direction gives essentially no response. So this is a cell that is interested in movement, and in particular in movement in a given direction. Having cells of this kind makes perfect sense in a visual system, because of course things that are moving in the world are of interest to the organism. If you're a rabbit, you're interested in things like hawks that might be moving. And so having cells that respond selectively to motion as opposed to simply the world that is unchanging has obvious selective value. And I think we can all appreciate that in our own everyday experience. For example, if a fly or some other object is moving in the peripheral visual field, that immediately catches our attention, and it does so in a way that non-moving objects do not. Now this brings us to a potential paradox. As we saw a few minutes ago, the head and body are generally moving around in space as we walk or go around our daily business. And even though there are compensatory eye movements, which work quite well to keep the eye largely oriented in space despite those movements, the compensatory eye movements are not perfect. And so the eye is constantly wiggling around back and forth in space, and therefore the image which falls upon the retina is constantly wiggling back and forth in the reverse direction. So if our retinas were composed of direction selective cells of the sort we've seen here, and these cells are the ones that are supposed to tell us what's moving out there in the world, we'd have a problem that these cells would be constantly stimulated simply by the motion that's referable to head, body, and eye motion, that is self-motion. What's the solution to that? It turns out that the solution is a yet cleverer set of cells, again, motion selective cells, but they respond only to local motion and not to global motion. And so, for example, if we deliver to one of these cells within its receptive field a grating of alternating black and white stripes which are moving from left to right, so they're just flowing across the visual field, the cell gives a very brisk response if that's all that is being presented to the retina. That is, the surrounding region of retina is being presented simply with a non-moving gray background. The cell loves this stimulus and responds vigorously. But what if we simply unmask the rest of the retina and show it the same set of bars moving from left to right? Now, the receptive field of the cell, this central circular region, hasn't changed at all in terms of the stimulus being delivered to it. Completely unchanged. The only thing that's different is that now the surround is doing exactly the same thing. What happens? The cell immediately falls silent. That is, this is a cell which wants to see a little region of the world moving against a background that is not moving. And if everything moves at the same time, that's not an interesting stimulus. This is perfect, because this is just the kind of cell that would tell a rabbit, for example, if a hawk were flying across the sky, but not if the rabbit had turned its eye a little bit and all the trees and bushes and everything else whose images are impinging on the retina had moved across the retinal surface. And of course, that's just what the rabbit wants to know. There's a very beautiful illusion, the so-called Ouchi illusion, named after the Japanese artist who initially derived this, which nicely illustrates this kind of motion-sensitive ganglion cells effect on our visual system. In the Ouchi illusion, and I've shown them here at two different magnifications because depending how you're viewing this lecture, one or the other might be more effective. In the Ouchi illusion, there are a series of horizontal black and white bricks which encompass the surrounding zone and are most of the image. And then in the center, the circular center, is essentially the same thing except it's been flipped vertically. And I think you can appreciate that the central region seems to sort of float around and bounce back and forth in a way somewhat different from the surround, as if it's almost autonomous from the surround. The origin of that motion is the spontaneous eye movements which we have all the time. Our eyes are constantly jiggling around in their sockets. And because of that jiggling, the direction selective ganglion cells are differentially activated by the center versus the surround of this illusion. So for example, if the eye were to make a horizontal excursion, a small horizontal excursion, these horizontal brick stimuli would be less provocative to those cells on average. Why? Because a small horizontal movement leaves much of the visual field with the same stimulus. The same little black zone that was stimulated when the eye was at one location will, if the eye now moves a tiny bit horizontally, still be stimulating in a black region of the new location. But the vertically arranged bricks will have a relatively larger effect for a given horizontal eye movement, because that little movement will be more likely to shift the gaze from, say, a black zone to a white zone, or a white zone to a black zone. And I think you can appreciate that the reverse is true for vertical eye movements. So that causes the visual system to make this segregation of the central circular zone from the surround, and it gives it this sort of bouncing back and forth, this sort of autonomy that grabs our attention and makes this illusion so effective. To continue with our theme of looking at how the visual system extracts information from the scene, we can ask whether having two eyes gives us some special advantage over, say, an organism that might have only one. And the answer is yes. Having two eyes does confer a special advantage. And in particular, it allows us to determine the depth of an object, that is, its distance away from ourselves, using a mechanism called stereoscopic depth perception. Let's see how that works. So if we have 2 points in the visual world, one here on the right and one here on the left, I think you can see by simple geometric optics how these would be imaged on the retinas of the two eyes, shown down at the bottom. And of course, the left point is imaged to the right on the eye because the image is reversed when it's imaged on the retina, and the right point is imaged on the left side. And those two images fall on what would be called corresponding points. They're corresponding points for the right spot, and they're corresponding points for the left spot. Okay, so there's nothing surprising here. But now let's ask, what if there is a pair of points that differ not by virtue of their position left and right, but by virtue of the fact that one is further and one is nearer to the viewer? Now, if you think about it, what the geometric optics tells us is that the innermost point is imaged further to the periphery on the left retina and also further to the periphery on the right retina. That is, the corresponding points for this closer object lie on opposite sides of the corresponding points for the further object. That's this more distant one here, which is now imaged on points that are closer in, more nasal, on the two retinae. Now that is a bit more confusing, perhaps, for the brain to figure out. What the brain has to do is figure out that the image that it sees for that nearer point on the left retina and on the right retina, those two images, are really representing the same object. This is the so-called correspondence problem. And because it falls on these different sides of that further object's image, therefore, it must be nearer. The nearer one can be detected as nearer by virtue of the fact that its corresponding points are lateral. And the further one can be detected as being further by virtue of the fact that its corresponding points are more nasal. Now that's a very difficult computational problem, as it turns out. It's not fully understood how it's done, but it's a very powerful system, and it gives us very accurate depth perception. As we consider further how the brain analyzes the visual scene, we eventually reach a point where that information is so complex that the analysis requires not just an assumptionless series of steps, but a series of steps that involve imposing some order related to our previous experience of visual information. Let me give you an example of this. Here we see a way in which we analyze depth from shading, and we have included in that analysis a hidden assumption. And the hidden assumption is that this three-dimensional object has been illuminated from above. That's a natural assumption because in general the sun, the classic illuminant, the classic source of illumination, is above. And so when we see an object, in general the shadows are below, and the rounded object can be analyzed in part based on the pattern of its shadows with that correct assumption that the sun is sitting above it. So here we have, just to illustrate this type of analysis, two images of cuneiform writing. In fact, these two images are the same image. The one on the right differs from the one on the left only by virtue of having been rotated 180 degrees. And yet I think we can appreciate that the one on the left looks as if the cuneiform is coming out at us from the plane, and the one on the right looks like the cuneiform is receding into the plane. And of course the reason is that when we see the shadows on the left, we immediately assume that the sunlight is from above, and therefore, since the shadows are below, the objects are coming out. And on the right side, the reverse. The shadows, again, if we assume the illumination is from the top, would tell us that the objects have receded into the plane. Okay, that's obviously a learned assumption. It's really an unconsciously learned assumption, and it's generally a correct one. Let's look at another way in which learned responses affect the way we analyze a scene. Of course, we've moved around in a world of three dimensions, and we're very tuned into the depth of three-dimensional scenes. We see some objects as closer, some as further away. In this little cartoon, we certainly get a strong sense that the walls form a zigzag pattern coming out and receding course, the gentlemen who are pictured here, by virtue of their size, reinforce that sense. And now if we look at these heavy black bars, the one that's here and the one over here to the left, we get a strong sense because we have assumed their different depths, that the bar that is to the right is a smaller object than the bar that is to the left. And of course, in the context of this three-dimensional scene, that is perfectly right. But in fact, if you measure the size of these two bars in this particular image, you simply put a ruler next to this bar and a ruler next to that bar, they're exactly the same size. Now, what's most striking is that once one knows that, even if you hold two rulers next to them and you prove to yourself that that is the case, you have a very difficult time convincing your visual system that that's true. That differential in size is a very strong and persistent sense. And again, it's a learned sense, and it is learned in the context of depth perception in the real world. Now let's look at one particularly famous example of this sort of imposing of order upon a scene. This is a commemorative vase that we're seeing here. It was made to honor Prince Philip and Queen Elizabeth. And I think you can appreciate that not only is it a beautiful white vase, but on one side is the profile of Queen Elizabeth, that is the shape of the vase makes her profile, and on the other side is the profile of Prince Philip. That's the nose right here, here's a nose right here. And the striking thing about the vase, as you view it and look for those profiles, is that the sense that it is a white vase, or the sense that it is two faces looking at each other, seem to be mutually exclusive. That is, you can see one or you can see the other, but it's hard to see them both or see them sort of fully and see them in a dominant way at the same time. And this is just an example, again, of the visual system imposing some order on the scene. It's trying to choose which of those two somewhat exclusive ways of looking at the scene is the correct one. Let's look at one final example. And this is a series of 4 sketches that Pablo Picasso drew of a bull, one of his favorite subjects. And I think you can appreciate that going from the upper left to the lower right, we're seeing an increasingly abstract version of a bull. Yet the striking aspect of this set of drawings is that despite the abstraction, we immediately recognize it for what it is. Now, like those simple line drawings, I think this begs the question why that should be so, why the visual brain is so good at identifying these sets of rather abstract images as representing a bull. And like those line drawings, I think our conclusion would be that what Picasso has hit upon is that these almost childlike drawings are really the internal representation of the bull, and that we have created a simplified and abstracted version in our heads, our memory trace of what a bull is, that really is this kind of drawing and therefore resonates immediately with it. Thank you.